Welcome to DSEJ Home Classes, an attempt to connect the learner with the instructor, a digital initiative by Directorate of School Education. Namaskar, my dear students. Today we are going to start with the chapter 6th of the class 11th, and the name of the chapter is the Anatomy of the Flowering Plants. And the topic that I will be discussing today is the tissue. So my dear students, when we talk about the anatomy of the flowering plants, I have to tell you that this chapter introduces you to the internal structures and functional organization of the higher plants. The branch of botany which deals with the study of the internal structures of plants and animals is called the anatomy. The branch of botany which deals with the internal structures of the plants is called as the plant anatomy. Now plants have cells as the basic unit, and cells are organized into tissue, which in turn are the tissues are organized into organs. Different organs in the plants show differences in their internal structure. Within the angiosperms, the monocots and the dicots are also seem to be anatomically different. Internal structures also show adaptations to the diverse type of the environments. And dear students, in case we want to study the anatomy of the plants, we must have study the tissues. As I told you, the tissue is a group of cells having a common origin and usually performing the common function. There are various types of the tissues in the plants, and let me introduce to some basic plant tissues. As you can see in this table, the plant tissues are broadly classified into meristematic tissues and the permanent tissue. Meristematic tissues are the tissues which have the power of division, that is, they can divide and add to the number of the cells. They are further classified on the basis of their position in the plant, on the basis of their origin. On the basis of position, that is where they are present in a plant, they are divided into apical meristem, intercalary meristem and the lateral meristem. Based on the origin, that is when they were formed in the life history of the plants, they are divided into promeristems or the primordial meristem, primary meristem and the secondary meristem. So we will be discussing all these types one by one, whereas the permanent tissue is one which has ceased the power of division, that is, it has stopped to divide and has got converted from meristematic tissue to the permanent tissue. The permanent tissues, they are again further subdivided into protective tissue, under which comes the epidermis and the cork, the supporting tissue, under which we shall be discussing the parenchyma, collenchyma and the sclerenchyma, and the conducting tissue, the xylem and the phloem. I must mention here that out of these, the supporting tissue is the simple permanent tissue, because this has only one type of cell in it, whereas the conducting and the protective tissue may be considered under the heading complex tissue, where they may contain more than one type of cells. We will discuss these tissue systems one by one, but to start with, we will talk about the meristematic tissue. Now, meristematic tissue, as already I told you, that these are further classified on the basis of their position in the plant body, that is where they are present in the plant, into apical meristem, lateral meristem and intercalary meristem. Whereas on the basis on the nature of the cells giving the meristem, they are divided into primary meristem and the secondary meristem. Now, what are the various characteristics of the meristematic tissues on the basis of which we can divide them, we can categorize them to be the meristematic tissues. They show basic anatomical and histological differences. The cells, as you can see in this figure, these are the cells of the meristematic tissue. These cells of the meristematic tissue are similar in structure and have thin cellulose cell wall. That is, all the cells, they are polygonal to oval to rectangular in shape, and they all have the cell walls which are thin and they are made up of cellulose. The cells may be round, oval, polygonal, rectangular in shape. They are compact and they do not have the intercellular spaces. What is intercellular space? That is, as you can see, the cells are closely fixed to each other. They do not have any space between the cells, and that space between the cells is called as the intercellular space, and that space is totally absent in the meristematic tissues. Since meristematic tissue is a tissue where the cells are actively involved in the divisions, and hence they have a large nucleus where constantly the chromosomes are under activity, and hence they have a large and conspicuous nucleus and abundant cytoplasm. The protoplasm contains very few or no vacuoles because the cells do not get the time to mature and develop the vacuoles. The cell undergoes division time and again, and hence the protoplasm contains either no or very few vacuoles. These are some of the characteristics of the meristematic tissue. Now let us discuss the various types of meristematic tissue. In this figure, this is the geometrical representation of the LS of the shoot system, which shows various types of the apical meristems. The meristems which occur at the tip of the root as well as at the tip of the shoot as well as the root, they are called as the apical meristem. The root apical meristem, however, occupies the tip of the root, while the shoot apical meristem occupies the distantmost region of the shoot axis. Now, during the formation of the leaves and elongation of the stem, some cells left behind form the shoot apical meristem and constitute the auxiliary bud. Such buds are present in the axils of the leaves and are capable of forming the branch or the flower. The meristem which occurs between the mature tissue, that is, this is the apical meristem. Now this white one is the mature tissue, and this represents the intercalary tissue, intercalary meristem. These occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristem and intercalary meristem, they are primary meristems because they appear early in the life of the plant and contribute to the formation of the primary plant body. Now there is the lateral meristem. The meristem that occur in the mature regions of root and shoot of the plant, particularly those that produce woody axes and appear along the lateral sides. These are the lateral sides of the stem. This is called as the lateral meristem. Generally, this is involved with the secondary growth of the plant. The apical meristem and the intercalary meristem, they aid to the increase in the length of the plant, whereas the lateral meristem increases the girth of the plant. The vascular cambium and intervascular cambium and cork cambium are the examples of the lateral meristem. These terms, vascular cambium, intervascular cambium and cork cambium, will be discussed when we discuss the secondary growth. However, I must mention here that these tissues increase the girth of the plant and thereby make the plant woody. Now, next is the permanent tissue. After the primary tissue, this meristematic tissue, we shall be discussing the permanent tissue. The cells of the permanent tissue do not generally divide further. That is, the permanent tissues have all the cells similar in their structure and function, and hence they are called as the simple permanent tissue. Whereas the permanent tissue having many different types of the tissue in one tissue system is called as the complex tissue system, the example of which is the xylem and the phloem. Whereas the simple tissue is made up of only one type of cell. The various simple tissues in the plants are the parenchyma, collenchyma and the sclerenchyma. We shall be discussing these one by one. Here you can see in the diagram the basic difference between the parenchyma, collenchyma and the sclerenchyma. There is the major difference is in between their thickenings of the wall. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal or elongated in shape. Their cell walls are thin and made up of cellulose. They can either be closely packed or they may have little intercellular spaces in between them. Now you can see the differences between these three types of the tissues. The parenchyma cells are relatively thin and the cells in the parenchyma tissue are loosely packed. Whereas the cells in case of collenchyma are irregularly thickened at the corner and there is very little space between them. As you can see in this diagram, they are thickened and the walls are thickened, leaving no space for the intercellular spaces. Whereas the cells of the sclerenchyma, the cell walls are uniformly thickened and there is not at all any intercellular space between them. The cell walls are usually uniformly thickened. Whereas in the collenchyma, they are thickened at the corners. Now, the cell walls in case of parenchyma is made up of cellulose, whereas pectin and amyloses are the major constituents of the collenchyma. In additional to all these constituents, in case of sclerenchyma, inwards the lignin layer is found and sometimes the layers of the lignin, they are arranged in the concentric fashion towards the inner side in such a way that they make the cell dead. Now these are the parenchyma and the collenchyma and the sclerenchyma. The tissue, there is the variation in the form of the sclerenchyma. The fibers are thick-walled, elongated and pointed cells, generally occurring in groups in various parts of the plants. The sclerids are spherical, oval and highly thickened dead tissue with very narrow lumen are the cavities, and they are commonly found in the fruit walls of nut, pulp of the fruit like guava, pear and sapota, the seed coats of the legumes and the leaves of the tea. Sclerenchyma provides mechanical support to the plant. Now let us discuss the permanent tissue of the complex type. These include the vascular tissue, the xylem and the phloem, and hence this tissue is also called as the vascular tissue. The complex tissues are made up of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissue in the plant. The xylem functions as a water conducting tissue and it carries the water and the minerals from the root to the stem and the leaves. It provides mechanical support to the plant. It has 4 types of different elements, and these 4 types of different elements are tracheids, vessels, xylem fibers and xylem parenchyma. However, gymnosperms lacks vessels in them. Tracheids are elongated or the tube-like structures. Here you can see the tracheids are the elongated and tube-like structures with lignified walls and tapering ends. These are dead and without any protoplasm. The inner layers of the cell's wall have the thickening which vary in form. In flowering plants, the tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube structure which are made up of many cells called as the vessel members, each with lignified walls and large central cavity. The vessel cells are also devoid of the protoplasm and they are interconnected through the perforations in their common walls. The presence of the vessels is a characteristic feature of the angiosperms only. The xylem fibers have this is generally star-shaped and have thickened walls and obliterated central lumen. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food material in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Now talking about the phloem. Now phloem transports food material usually from leaves to the other parts of the plants. The phloem in the angiosperms is composed of sieve tubes, companion cells. These are phloem parenchyma and the phloem fibers, which is also called as the bast. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and the companion cells. The sieve tubes elements are also long tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in sieve-like manner to form the sieve plates. A mature sieve element possesses the peripheral cytoplasm and a large vacuole but lacks a nucleus. The function of the sieve tubes is controlled by the nucleus of the cells, which are called as the companion cells, which are specialized parenchymatous cells and are closely associated with the sieve tube elements. The sieve tube elements and the companion cells, as you can see in this figure, the sieve tube elements and the companion cells are connected by pit fields present between their longitudinal walls like this. This is the pit field where they are connected with each other by the longitudinal walls in between them. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering, cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and have pits through which the plasmodesmatal connections exist between the cells. The plasmodesmatal connections are the connections between the adjoining cells through which they can communicate with each other. The phloem parenchyma stores food material and other substances like resins, latex, mucilage, etc. Phloem parenchyma is absent in most of the monocotyledons, and phloem fibers or the bast fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem. Now, on the basis of whether the xylem or the phloem is formed first or the later, we have divided the phloem into or the xylem into two types. For example, here you can see protoxylem and metaxylem. The protoxylem, the first formed xylem, and the metaxylem means the late formed xylem. Protoxylem exists for short period of time, after which it gets converted into the metaxylem and then it prevails forever. Fibers are absent in protoxylem but get develops during the formation of the metaxylem. Protoxylem cells are narrow in diameter and they are broader in the metaxylem. Now you can see in case of TS of dicot stem, the metaxylem is towards the outer side and the protoxylem is towards the inner side. In case of root, the protoxylem is towards the outside and metaxylem is towards the inside. When the protoxylem lies towards the pith and metaxylem lies towards the periphery, this type of condition is called as the Endor condition. In roots, the protoxylem, as you can see here, the protoxylem lies towards the periphery and metaxylem lies towards the center. Such a arrangement of the xylem is called as the exarch condition. Now that's all about the different types of the tissues, that is the primary tissue, this is the meristematic tissue and the permanent tissue, among which they are the two types, the simple tissue and the complex tissue. Next time we will be talking about the tissue system and the secondary growths. Till then, thank you, stay safe, and if in case you have any question, you can ask me on my phone number. God bless you. Thank you so much.